Common depictions of human fertilization often resemble a microscopic battlefield. We see millions of sperm racing toward an egg, appearing as biological drills attempting to force their way inside. From a biochemical standpoint, this model of cellular invasion is inaccurate. Cells do not achieve entry by simply bashing against physical barriers. A sperm cell unleashing generic tissue-dissolving acids to melt through its target would be counterproductive. The resulting chemical damage would destroy the egg cell entirely. The egg is surrounded by specialized extracellular layers, complex structures that regulate sperm interaction through specific chemical signaling. Navigating these defenses requires one of the most precise molecular exchanges in biology. Successful entry relies on coordinated recognition rather than physical destruction. The egg is protected by cumulus cells and a dense glycoprotein shell, the zona pellucida, above the plasma membrane. To deliver its genetic material, the sperm navigates these barriers using the acrosome, a specialized organelle at the front of its head. While the acrosome shares evolutionary origins with lysosomes, it functions as a highly specialized tool adapted specifically for fertilization. It carries targeted enzymes, such as acrosin, designed for regulated interactions with the egg's protective coatings. The sperm utilizes this specialized enzymatic toolkit, precisely calibrated to navigate the unique chemistry of the egg's protective layers. The process begins the moment a sperm makes contact with the egg's environment, triggering the first step of molecular recognition. This contact initiates the acrosome reaction. The sperm head membrane alters, releasing its enzymatic payload into the surrounding space. These proteins engage the extracellular matrix, modifying its structure, allowing the sperm to part the layers without damaging the egg. This precise enzymatic release alters the local environment just enough to allow the sperm to continue its physical journey forward. Targeted enzymatic activity serves strictly as a facilitator for movement, ensuring the mechanism for entry is based on chemical interaction. After navigating the extracellular layers, the sperm reaches the final barrier, the egg's plasma membrane. Here, molecular players Izumo-1 and Juno act as a biochemical lock and key. These proteins extend from their respective membranes and interlock. This successful handshake signals mutual recognition, causing the two distinct cellular boundaries to adhere and merge. Membrane fusion allows the sperm's genetic material to cross the final threshold and enter the egg. Ultimately, at its core, the process of fertilization is a cooperative act of mutual cellular fusion and acceptance. Fusion creates an immediate biological challenge. If a second sperm were to enter, the resulting genetic imbalance would halt development. The egg responds with the cortical reaction, a rapid defense mechanism triggered the instant the first cell fuses. This reaction chemically modifies the zona pellucida, creating an impenetrable block that prevents any remaining sperm from entering. The egg is an active participant in this process, instantly securing its borders to protect the new genetic pairing. This microscopic choreography has significant real-world implications for human health. Infertility often results when any part of this sequence, from the acrosome reaction to the protein handshake, fails to execute correctly. By decoding these molecular mechanisms, researchers can develop more effective assisted reproductive technologies to help overcome these biological hurdles. Human development relies on this million-year-old molecular code. It confirms that life begins with highly coordinated biochemistry.